Welcome to this explainer. It's kind of wild to think about, but the everyday act of just swallowing a simple pill relies entirely on invisible, microscopic geometry. We're going to connect that incredibly common experience of taking medication all the way down to the tiny cellular architecture that actually cures us. Because ultimately, this is a story of engineering, but on a molecular scale, where the precise angles of atoms literally dictate human health. So think about this for a second. How does a pill know exactly where to work in your body? You pop a tablet for a headache or maybe a twisted ankle, and it has to navigate a ridiculously chaotic environment. We're talking about traveling through your digestive system, metabolizing in the liver, and pumping through kilometers of blood vessels. Yet somehow, amidst all that biological noise, it manages to find the exact source of your pain. It doesn't just shut down your entire nervous system, right? It targets the specific problem. Unraveling that biological mystery means we have to look closely at the geometry of healing.
Okay, let's dive right in with part one, the macro. What are drugs?
Fundamentally, drugs are just chemical substances deployed to prevent, diagnose, or treat diseases. And that covers everything from your standard daily vitamin all the way up to highly advanced oncology therapeutics. But we have to remember that a chemical substance is essentially inert until it physically interacts with your biology. The substance itself isn't the cure. The cure is what happens when the geometry of that substance meets the geometry of your cells. Drugs don't just float randomly around your circulatory system hoping for the best. They are designed to act on incredibly specific destinations. We call these targets, things like enzymes, proteins, nucleic acids, and entire cellular pathways. Just think about the sheer scale of this for a moment. Your body has trillions of cells, and each one contains thousands of different proteins. When you take a medicine, its chemical components are on a highly specific mission. They bypass billions of incorrect destinations just to seek out and interact with one precise bodily target to create a physical change. That brings us to part two, the metaphor, the lock and key. To really grasp how these chemicals interact when they finally reach their destination, we have to look at the bioactive molecules inside the drug that actually do the binding. These are known as ligands. The ligand is the active agent doing the heavy lifting. It meets a receptor, which is typically a complex protein situated on or inside your cells that is totally primed and ready to receive and bind to this very specific ligand. The lock and key metaphor is an absolute classic way to think about this specificity. The biological target, the receptor or the enzyme, is the lock. The ligand, the bioactive molecule in the medicine, is the key. But while it's a super helpful analogy, the reality is much more nuanced than just physical shape. It's fundamentally a story of spatial geometry and molecular forces perfectly aligning in three-dimensional space. The shape of a drug's ligand absolutely must align with the incredibly complex topology of the target's active site. In medicine, a perfect fit means a strong, effective interaction. Just like your actual front door, the exact arrangement of grooves and ridges dictates success. But what happens if the geometry is slightly off? Well, a wrong key doesn't just fail to open the intended door. In the bustling metropolis of the human body, an incorrectly shaped molecule might accidentally fit into the wrong receptor, opening the wrong door entirely. And this is precisely what causes unwanted side effects. The geometric shape of the molecule is the absolute crux of safe, effective drug action. Now for part three, the VSEPR model. So if shape is so universally critical for a drug to work without causing massive side effects, how in the world do chemists know the shape of a molecule before they even physically synthesize it in a lab? I mean, we are talking about structures measured in nanometers here. They are far too small to see with the naked eye. Designing a microscopic key for a biological lock we can barely even observe is just an incredible engineering challenge. The solution to this whole challenge lies in VSEPR. That stands for valence shell electron pair repulsion. Yes, it is a bit of a mouthful, but it's a remarkably elegant concept that totally dictates the rules of molecular architecture. The VSEPR model is basically a super reliable, predictive tool that allows chemists to determine the precise three-dimensional shapes of molecules. And it relies on a very simple physical reality. Electrons have a negative charge, and like charges repel each other. Because of this, the electron pairs surrounding a central atom will naturally arrange themselves as far apart as physically possible in three-dimensional space, just to minimize that repulsion. By mapping out these repulsions, chemists get a flawless blueprint. They can accurately predict the exact architectural structure that a collection of atoms is inevitably going to form. This predictive power literally transforms medicinal chemistry. Instead of relying on blind trial and error, mixing chemicals for years in a lab just to see what shape they might randomly take, the VSEPR model helps scientists design drugs with the correct geometric configurations right from the start. It allows them to recognize biological substrates and mathematically map out exactly how those structures fit together. It is deeply fundamental for understanding ligand and antibody recognition. Because they can accurately predict this spatial geometry, scientists can custom cut the molecular key to ensure it will seamlessly and safely slide right into the targeted biological lock, which ends up saving years of research time. Now, what's really interesting is how we bring this back to reality. Part 4, the application. Aspirin in action. Let's apply all this microscopic geometry to a medicine that almost everyone has interacted with at some point: aspirin. Aspirin is over a century old, but it actually wasn't until modern chemistry matured that we fully understood its geometric interactions at a molecular level. It works by targeting an enzyme known as COX, or cyclooxygenase. In our analogy, the COX enzyme is the lock. So here's how it plays out. The aspirin molecule contains an acetyl group, which acts as our ligand, our key. Because of its highly specific three-dimensional shape, which is perfectly predicted and mapped through the rules of the VSEPR model we just talked about, this acetyl group binds exactly to the active site of the COX enzyme. This precise geometric fit acts basically like a physical wedge jammed in a door, stopping the COX enzyme from functioning at all. And by stopping that specific enzyme, the biological cascade of signals that cause inflammation and pain is immediately halted. The VSEPR model explains the very physics of why this binding is even possible. It all comes down to exact, mathematically predictable shape compatibility. Understanding the shape of molecules is clearly not just some abstract chemistry exercise. It is the absolute foundation of modern pharmacology. The VSEPR model allows us to design incredibly target-specific drugs. It grants us a profound understanding of binding interactions at the atomic level. And ultimately, this geometric precision is what improves the effectiveness and the safety of the medications we rely on every single day. When the geometry of the key is perfect, the drug works flawlessly, neutralizing the problem while leaving the rest of the body's incredibly complex machinery completely undisturbed. We've seen exactly how the precise geometry of something as simple and common as aspirin acts as a molecular key to just turn off